So, die letzten Gäste sind noch am Counter, aber kommen auch gleich rein. Dann können wir nämlich fast pünktlich mit unserem Programm starten. Ja, liebe Freunde, Freundinnen der Fotografie, liebe Mitglieder des Freundeskreises, des Hauses der Photographie, liebe Gäste, dear guests and friends of photography, herzlich willkommen. Welcome. Also, ich switche noch ein bisschen zwischen Deutsch und Englisch. Wir werden nachher ins Englische wechseln. Meine kurze Begrüßung werde ich auf Deutsch halten und wollte mich jetzt am Anfang erst einmal bedanken, dass ihr alle gekommen seid, dass trotz aller widrigen Wetterbedingungen und Bahnbedingungen die Referenten da sind, die Referentinnen da sind, dass fast alle auch allen Viren getrotzt haben und heute hier bei uns sind und wir mit unserem Jahressymposium starten können. Also wie schön, dass ihr alle wieder dabei seid. Ich danke euch natürlich allen, dass ihr unsere Arbeit des Vereins unterstützt, dass wir überhaupt solche Formate wie heute möglich machen können. Und wir freuen uns natürlich umso mehr, wenn unsere Überlegungen dann auch ein so großes Interesse stoßen und das Auditorium dann wieder richtig voll ist. Unser Jahressymposium ist ja immer ein Höhepunkt in unserem Programm. Das bedarf natürlich auch immer einer besonderen Vorbereitung, hat sich aber eben traditionell schon sehr gut bewährt. Es ist immerhin die 14. Folge von Jagen und Sammeln. Also das ist tatsächlich ein Langzeitformat, ein Longtime-Runner und es ist immer wieder spannend, wie sich dann im Laufe des Jahres vielleicht auch die Ideen entwickeln, was wir denn am Ende des Jahres zu unserer Sammlerstücke-Präsentation auch als wissenschaftliches Programm oder als Vortragsprogramm anbieten wollen. Wie immer nehmen wir eben bei unserem Jahressymposium bemerkenswerte Fragestellungen der Fotografie ins Augenmerk und schauen natürlich auch, was im Haus der Fotografie läuft und da war es natürlich naheliegend bei der wunderbaren Ausstellung von Katrin Linkersdorf, diese Ausstellung zum Anlass zu nehmen, uns mit ökologischen Fragestellungen der Fotografie näher auseinanderzusetzen. Also, gemeinsam haben wir das Programm entwickelt und ich möchte vielleicht an dieser Stelle, weil es ganz am Ende immer untergeht, muss ich das jetzt gleich am Anfang machen und ich will es am Anfang machen. Das ist mir eine große Ehre, auch als Vorstandssprecher mich bei meinem tollen Team auch zu bedanken, dass wir hier gemeinsam diese Veranstaltung entwickelt haben. Also es gibt immer die sichtbaren Mitglieder des Freundeskreises, aber es gibt eben auch die unsichtbaren, die hinter den Kulissen ganz viel für uns vorbereitet haben. Also ich danke dem Team und möchte da namentlich vor allen Dingen, also ich bedanke mich bei meinen Vorstandskollegen, ich bedanke mich aber jetzt ganz explizit bei unserer Geschäftsführerin Ulrike Klug. Das ist wirklich ein wahres, unverzichtbares ein unermüdliches Organisationswunder. Ich muss das wirklich sagen, am Ende klappt immer alles und wir sind hier wirklich dann ganz entspannt. Ebenso danke ich natürlich Sonja Schepke, die mit großem Engagement unsere Mitgliederbetreuung organisiert und die natürlich auch hier zusammen mit unserer Kollegin Annette den Einlass organisiert hat. Also ich danke allen aktiven Okay, so viel Zeit muss sein. Ich danke allen aktiven Mitgliedern des Freundeskreises des Hauses der Photographie und ich danke vor allen Dingen meinen beiden Mitstreiterinnen, die dieses Symposium hier organisiert haben mit mir, Stephanie Bunk und Cale Garrido. Die werden Sie ja gleich nochmal ganz genauer kennenlernen. Cale Garrido ist neu im aktiven Kreis des Freundeskreises. Deshalb möchte ich Sie ein bisschen näher vorstellen. Stephanie Bunk und mich, Ulrich Rüter, kennen Sie ja schon lang genug, sage ich mal. Aber Cale ist in Hamburg durchaus ja auch keine Unbekannte, wenn man an die Triennalen der letzten beiden Triennalen denkt oder auch an das Raw Photo Festival in Worpswede, wo wir sie ja auch besucht haben. Und Sehr gerne gebe ich das Wort gleich an Sie ab, darf Sie aber noch kurz vorstellen und dafür wechsle ich tatsächlich jetzt auch schon ins Englische, damit wir uns alle daran gewöhnen, was unsere 3 Referenten uns dann hier heute Nachmittag präsentieren werden. Und eine Ansage muss ich nochmal machen, ich habe es schon drüben bei der wunderbaren Führung mit Inge und Katrin gesagt, es wird gefilmt und es wird fotografiert. Also wenn jetzt jemand nicht sichtbar sein möchte, muss sich entweder eine dunkle Mütze aufsetzen oder sich ein wenig zurückhalten. Aber wir wollen natürlich diese Veranstaltung auch nutzen, um dann für alle, die jetzt dann doch zu Hause im Bett liegen müssen oder es nicht geschafft haben, auch später nochmal etwas zum Gucken haben und auch zum Hören und Sehen haben, weil diese 3 Vorträge, die wir heute hören und sehen werden, sind, glaube ich, sehr spannend und ich freue mich sehr, dass die Referentinnen dann auch wirklich alle da sind. Also, jetzt darf ich Cale Garrido vorstellen. She's a Spanish independent journalist, curator and a photo editor based in Hamburg. You all know this. After her journalism studies, she worked as a photo editor, was curator of several exhibitions and was a curatorial team member at Raw Photo Triennale in Worpswede. And das war in 2023, and the 8th Triennial of Photography in Hamburg last year. And she will give us now a deeper insight into the questions we asked ourselves in preparation for the symposium and what we can expect in this afternoon. So, Cale, you have the stage.
So I'm going to share the program here so you have it in mind, what is expecting us in the next 3 hours. Vielen Dank, lieber Ulrich, and thank you very much. I'm going to switch directly to English. Good afternoon, everyone. Ulrich has mentioned that this is the 14th Jagen Sammeln-Symposium. However, it is only my second event since joining forces with my colleagues at Freundeskreis. I am delighted to address you, dear audience, on this occasion, bringing up this particular subject and alongside very special guests. So, picking the right words is key to framing a topic, be it for an article, a book, an exhibition, a symposium, a scientific study, or an artwork. Many of us face this challenge on an almost daily basis. I have to say it wasn't easy to pick the title for this symposium because dear Boaz Levin took the best words for the 2 exhibitions he did. So yes, we negotiate with colleagues and with ourselves and ask, how will specific words affect us intellectually and emotionally? And will certain words or terms be more engaging or turn people off? From where I stand, it is tough to choose the right term to speak about what we often refer to as climate crisis, a concept that encapsulates a variety of interlinked emergencies, such as global warming and pollution, ocean acidification, mass extinction, you name it. So can we agree on a single word when the events we refer to are massively distributed in time and space and exceed our human scale of perception and comprehension? As with words, can we think of an appropriate visual language that can both represent the climate crisis and raise awareness? What is the potential of a photographic image beyond capturing a moment in time or at specific location? Today, we might not want to claim a single strategy, nor give simple answers. I like what writer and critic T.J. Demos says about this. The diversity of approaches is a cultural mirror of biodiversity itself. This describes the essence of our intention this afternoon, and when I say our, I speak for the Freundeskreis and for my colleagues Stephanie Bunk and Ulrich Rüter, by gathering thoughts and minds around science, artistic practice, and curating. So what does it mean to think of an image, of an exhibition, of a book, from an ecological perspective? Is it really just about reducing waste and carbon footprint? What role do mediation and accessibility play here? Is there such a thing as renewable photography? Or, as Taco Hidebaka puts forward, is image making a handmaiding of global warming, or could it be a tool for cooling too? So traditionally this conference takes place once a year ahead of the winter break. And as we learned that Katrin Linkersdorf's works would be on show at this time of the year, our idea to address the ecological aspects with photography and image making started resonating even more deeply within us. Not only does she work at the intersection of photography and botanics, as some of you might have seen in the exhibition, but through her recent research, she has also drawn our attention to her collaboration with microbiologist Professor Regine Henge from the Humboldt University in Berlin. We are very thrilled she's here as a speaker today. And I ask myself, beyond scientific data and evidence, what other narratives can the microscopical lens inspire? And what can be seen through bacterial performance? It appears that our human world isn't the only one alive with intelligence and agency. What a surprise. So speaking of interdisciplinary and collaborative practice, let me introduce our second guest, Mathieu Asselin. In short, his work is based on long-term investigative research, but it has so much more. I first met him in 2017, just before the release of his project focusing on the agricultural corporation Monsanto. Since then, I always sensed that this, his first very well-known photographic investigation, would have a continuation. And as in nature, everything has its own time. So lately, in a conversation following the invitation to this conference, it came as no surprise when he mentioned his ongoing project. While it's not my intention to anticipate his lecture, and actually I cannot because it's the very first time he does this lecture, I would like to foreground his genuine curiosity and persistence. Following the thread of companies, commercials, and propaganda, his latest work links photographic, archival, and material research, shedding light on power structures and the dubious logics behind capitalism. And it is precisely this same logic that underlies the means of representation. So consider any analog, any digital image, what resources are necessary to produce that image, and for that image to circulate. How do these techniques relate to energy, to labor, to waste? How does the history of photography intersect with the social ecological crisis that we face today? We couldn't think of a better interlocutor to discuss or even to practice self-reflection within the realm of the photographic medium than Boaz Levin. So he's our next speaker, and his lecture, as you can see, titled Beyond Fossilized Sunshine on Photography's Metabolic Turn, he will guide us through some of the main ideas of his curatorial research and present a selection of artists he has collaborated with. So let's begin. Boaz, I give you still a few sentences. He's a writer and curator based in Berlin who explores relations between politics, technology, and the environment. He was co-curated at the Biennale für aktuelle Fotografie in 2017 and the Chennai Photo Biennale in 2021. But now I will mention what some of you might already know. Last year, he curated together with Esther Roels Mining Photography, the ecological footprint of image production at Museum für Kunst und Gewerbe here in Hamburg, which later traveled to venues in Vienna and Winterthur. And also, together with Katrin Schöneck, he's presenting a continuation of his research on image ecology. This exhibition is still on show at the Oberlin, I think, until the beginning of January. And it's exactly this institution where he is now co-head of program and curator with Sophia Greif. So, dear Boaz, the stage is yours.
Thank you so much, Cale, and thank you so much to the Freundeskreis. It's really, truly a pleasure. Let me know if I'm speaking too loud or too quiet. Everything's okay? Everyone in the back? Good. If there's anything, just let me know. And special thanks, of course, to Stephanie and to Ulrich, and as I said, to Cale for this generous invitation and this chance to explore a couple of recent projects. What I thought I would do today is a bit ambitious because I know myself, and usually, so I'm going to talk about two recent exhibitions, one of which I hope maybe some of you would have seen here in the Museum für Kunst und Gewerbe, and the other is the show which is currently on view, as Cale mentioned, in CO Berlin. And I'm going to try and kind of talk about both. Usually talking about one takes me way more than an hour, so I knew when I thought that this is what I'm going to do, that this sounds like an interesting proposition which is worth exploring but might fail. But I'm going to keep it short in any case. And I think the reason I want to talk about both is that for me this was really a process, a learning process, an experiment, a process of discovery. And one project led to the next. And what I want to share a bit today is some of the thoughts, let's say, that I discovered during this process and some of the questions that it raised for me. So I think, yeah, maybe all of us together can later, think about how these things work together and what are the differences during the questions and answers. Wait, this is a bit of a strange position. So, yeah, exactly, it's important. It's quite warm here. So, Beyond Fossilized Sunshine on Photography's Metabolic Turn is the title of the talk. What is fossilized sunshine? I'll talk about this shortly. My starting point for both of these projects was in around 2018. I was asked to write a short text about for the Wüstenrot documentary photography prize for the catalog about what, and the prompt was really to write about what I think might be the key challenge for young documentarists or for artists working with photography who think about documenting their world. And what came to mind for me at the time, and has stuck with me ever since, is that, of course, the climate change is for me, I don't know about you, but I think for me it's something which I've been thinking a lot about and struggling with and trying to reckon with, and seemed like maybe the key challenge of our times, not just for artists, but maybe especially for artists who are trying to work to document their environment, to document the crisis of their environment. And the reason, I think, so I tried to break down in that text, and for me that moment was like a first foray into this discourse, into these questions. And what I realized was that there's a certain paradox kind of baked into our interaction with climate change as a subject. It's, on the one hand, omnipresent, but it's also very abstract, hard to grasp. We, of course, have become, it's horrible to say, but kind of horribly used to a certain type of image which comes to stand for the climate crisis. I've taken this image because this was the image that's stuck in mind for me in 2018 when I started writing about this subject. It was the burned remains of a city called Paradise in California, one of the worst fires at the time. And of course, when you start writing about this every summer, you start enumerating the records, horrible sort of records that are being broken with every wildfire, which is larger than we try to grasp for superlatives, to describe the new horrors we're confronted with. But what's interesting about this, so on the one hand we see these images, we see images of melting glaciers, of fires, of wildfires, of the last summer smoked out skyscapes, let's say in New York. But despite seeing these images, these are images of the effects of climate change rather than the causes. So what we primarily see are often these type of images, kind of sublime sense of sublime catastrophe shot from above, from a bird's eye view. And it tells us very little actually about how we got here, and it's often, in a sense, distant. If we think about the trope of the sublime, if any of you have studied art history, this goes back to, it's a trope that emerges, key to Romanticism, emerges in the 18th century, 19th century, crystallizes, and it's very much about this kind of pristine, giant, immensity of nature and the human being small and actually lacking agency vis-a-vis what is represented. So also this is interesting in the sense that the type of images that reach us are very much images which imply that these are almost, we could say, natural catastrophes. They are larger than us, distant, far away. This is not only something which is common in photojournalism, an iconic and I think important exhibition. And again, when I'm talking here, kind of the criticism I have of photojournalism, I should say, is not, I very much subscribe to what Cale said. I think there are a lot of strategies we should employ. But my perspective is one which I thought was kind of maybe not considered. So I wouldn't make this too personal about this or that approach. But there was this exhibition, which was very important at the time, Anthropocene, showing work by Edward Botensky, most famously. And this is, again, very similar types of images of large landscapes shot from above. And so when I started thinking about this, I kind of noticed, there's this tension. It's difficult to reckon with when the more you start actually reading about the history of climate change, about its origins, and let's say about what people now describe as the Anthropocene, other people describe it as the Capitalocene or fossil capitalism, the age in which humans have become the most important variable on our climate systems, on our planetary systems. When you read about that history, one of the things I realized was, so this is usually the graph that comes up. It's called the Great Acceleration. It shows how all our planetary systems started going whack, started, accelerating. That's why the Great Acceleration. At a certain point in history, it's very, very, oh no, it's not so small here, it's good. So you can see when is this happening, roughly 1830. What else happened around 1830? First photograph, 1827. So this really coincidence was the origins for the project I curated here, Mining Photography at Museum für Kunst und Gewerbe. The starting point was kind of trying to make sense of what might ostensibly seem like a negligible historical coincidence. But once you start digging deeper, you realize maybe it's not so much of a coincidence, maybe it can actually teach us something, both about photography and about the relationship to the environment more generally. So, what would it mean to tell a history of photography, which is also a history of the environment? What I call environmental history of photography in some of the recent texts I've written. This image, which most of you, all of you know, this is the Freundeskreis of the House of Photography. The image of the roof that Niesifor Niepce shot from, next to the Saône River, the point of view. It's actually a heliograph, he describes it, on a pewter plate. This is considered the earliest lasting, so surviving photographic image. And that's important. It's an image that remained fixed. And what is little known is that I don't know how well you know about this history, but what is often not so often discussed in photographic circles is that Niepce was actually busy for most of his first 20 years working on a very different invention. He was dedicating most of his resources and energy with his brother, working on something which they both thought would completely change the world. And they were right. Although they gave up this invention, they are considered one of the key inventors, I mean, in a way, maybe the most direct predecessor of today's internal combustion engine, which you have in most automobiles. What happened was they submitted a patent, and after a while they had a lot of technical challenges. They managed to actually power a boat up the Swan River, not so far from where Niepce would take his photo, a boat of 900 kilos against the stream. But in the end, Claude, his brother, died. They ran out of money, yada, yada, yada. Niepce went on to invent photography, which was his side project. But what's interesting, actually, in the invention of photography, when looked at through this lens, is that the key element, those of you who've read about the invention of the heliogravure, what actually Niepce's breakthrough was his ability to fix the image. There were a lot of inventions, camera obscuras, different techniques, methods being attempted at the time. And the key breakthrough for this was his realization that bitumen, which is raw petroleum or asphalt, when it's exposed to light, it becomes non-soluble. It doesn't dissolve, so it can be fixed. So in that sense, you could say, when we look through this lens and look at the history of photography through this lens, the first image was literally etched in fossil fuel. One theoretician who wrote for a book, and whose book has been really invaluable for us, Nadia Bozak, described it as fossilized sunshine. And fossilized sunshine is actually a term she took from a very important environmental historian who uses this term to describe oil, because that is oil. It's solar energy decomposed over millennia under specific conditions, and then it can be accessed again, tapped into again. And when you think of histories of the environment, really what people point as the turning point for the great acceleration is usually the introduction of steam, coal-powered steam engines. This is what is the great kind of multiplicator of both of efficiency, but also, fossil capitalism. That's why it's called that way. So how did you tell this type of story? Just gonna drink some hydrate. That was our challenge in this exhibition. Some of you might have seen it, so I won't really talk about it too much in depth. I'll speed through both exhibitions. As I said, it's too ambitious, what I'm trying to do, or too optimistic. Mining photography was our attempt to tell the story, to tell a different history of photography, which in a way looked at everything that usually is left outside the frame, So, as you mentioned, the labor, the resources, the waste that's left after a photo is taken, these are things that, if they are taken into consideration in the history of photography, they are seen as incidental, mostly, throughout its history. And we can talk a bit about why this is the case later, like the ideology of the project of photography. But how does one do this, and what does this mean? So the first image one saw when entering the exhibition was this image from an archive I found in the Kodak's archive in Rochester. This looks like a kind of Ocean 11 safe, and it actually is a safe with very expensive amounts of silver. And I'll just show you the verso from the archive. This is 1945. These are silver ingots in Kodak's safe, and at the time Kodak had the second amount of, the amount of silver in Kodak's safe was second only to the US Federal Reserve, which means basically it was second in the world. So this was kind of the entry point for our exhibition, but the exhibition was structured chronologically, and we took key materials in the history of photography. I have to admit, somewhat randomly, but what guided us was the artifacts from the museum's collection. So each chapter had 3 levels, basically: an item from the museum's collection, commentary by some sort of scientist or commentator, theoretician, about this item, kind of trying to shed a light from a very different non-art historical perspective, mostly, or materialist perspective, and contemporary artworks, responding both to which deal in a way with the traffic of this material. As you see, some are actually not one material, but 2. We can say something about these choices. But so basically, yeah, silver, the reason you entered and saw it is because silver is, of course, the key material in the history of photography to this day, and because in silver you can really see the crystallization of the photographic industry as a key consumer. So photography by 1999, the photographic industry was responsible for 60% of the global consumption of silver, which is, yeah, I would not have imagined that when I started working on this project. So those are the kind of things we learned and discovered and were surprised by. I thought as an example to how the project worked, as I said, I'm not going to go through all of the works, definitely not, and also not through so much through the chapters very rigorously, but I wanted to focus on the example of copper, because it's connected to Hamburg and because it's kind of a good example to show how we worked and how we researched in the exhibition. So copper stands for the daguerreotype. The starting point, the oldest object we had in the exhibition was a daguerreotype. Daguerreotype is one of the kind of oldest successful mass media in the photographic history, although it's exactly on the cusp of this artisanal and industrial, let's say corporate phase in the history of photography. And we had the pleasure of being able to work with this amazing image from the museum's collection, which is a daguerreotype from 1847 by Hermann Biot. I forgot to write that down, of a certain anonymous figure in the history of science and German thought, Alexander von Humboldt. And Humboldt, for us, this was a really interesting moment also to think through kind of how you can look at photography differently, because Humboldt stands for a lot of things. Humboldt as a subject, as a person, as a historical figure, stands for a lot of things. He is well known, of course. There was a book called The Invention of Nature that was a huge bestseller a couple of years ago, which described him as one of the first people to identify human-made climate change, anthropogenic climate change in South America. He was, of course, a maverick, a polymath. What he also was a mining officer. A mining officer and a mineralogist, avid mineralogist. He collected mineral samples from wherever he went, from the Russian Caucasus to South America. And in this image, we kind of wondered, could we, or speculated, could we find out where the copper came from in this daguerreotype, and what would that teach us about the history of photography and also maybe about the history of trade at the time? This is a copper copper plate in Hamburg being taken by a photographer. And we had this, maybe a bit this fantasy that could it be that the copper might come from the same locations that Alexander von Humboldt traveled to, Potosi, copper and silver. So what we discovered during this process, I won't go through the whole process, it was very complicated. And no one knows anything about where, there's very little information about the production of copper daguerreotypes in the 19th century. As I said, it's this moment of transition between like artisanal, industrial, and at the time, yeah, there are no files, like if you go to AGFA, there's no archive that you can access. So we tried different ways that didn't work. We tried identifying the hallmark. And in the end, what happened was that we worked a bit like the conversation later with a scientist. We thought what we could do is go to try and approach an archaeometallurgist, because in archaeology, for those of you who are nerds like me, you know that there's a lot of studies about the provenancing of metals, of coins, let's say. It's a very important field, archaeometallurgy, where you can study try and establish the provenance of a metal, let's say of a Roman coin. If you establish that it might have come from India, this is invaluable to understanding of civilization at the time. So this is something that exists. You use what's called a lead isotope signature. You try to identify it's an isotope analysis, lead isotope analysis, which can be traced more or less roughly to specific areas or deposits. It's still very rough, not so specific. And we went through all of this kind of goose chase to try and understand where the metal might have come from. What we discovered is that it might have very well been from Wales. Wales, the UK, which became in the 19th century a center of global copper production. So already you see the other option, which is a bit less interesting, would have been the Harz, just to give you the full disclosure. But you already see the kind of, both the maybe experimental means one has to use in order to try and answer these questions, but also the type of answers you have, you find out through this study. So why was it Wales? Wales became a global center of copper production, not because of its copper deposits, but because of its coal deposits. No, it's access to coal. So copper would be shipped to Wales in the 19th century and processed because of coal. Ergo, you could say photography, the daguerreotype would need this kind of industrial and global network already in one of the first media or first surfaces where it emerges. As I said, I'm not going to talk about all the projects. Hopefully you've all seen. Yeah. Yeah, exactly. Okay, so this, but what I wanted to, the reason I wanted to talk a bit longer about the copper project is because you see the method. And the method and the way we approach the artifacts we had. And this is something that then we repeated throughout the different sections in different ways. As I said, we had coal and bitumen. And again, this included both artistic positions and artifacts from the collection. And I don't want to talk about them. I just want to go through them so you can see the scope of the exhibition. Because hopefully, yeah, we're in Hamburg, so it doesn't make so much sense. This was silver. As I said, silver is kind of the main key material. We got to paper and its coating, cotton, pulp, gelatin, celluloid. Really interesting history, both in the aspects of the history of cotton and paper, which we really, I think, most of us take for granted, but in the 19th century, most of the photography paper was cotton rag, and combination of cotton and flax. Cotton, the biggest producer of cotton in the 19th century was the US. The reason it was the biggest producer was its reliance on the sharecropping system and before that on slavery. And copper also, cotton has a really interesting history because it connects to the figure of the rag picker, the people who are collecting rags in the 19th century, which was mostly children, women, the urban poor, which were kind of the quintessential figure of 19th century, the proletariat. So no, the origin of the word proletariat, a Lumpenproletariat is from Lumpen in German, Lumps is rags. So as you can see, the different materials tell different aspects of a similar history of labor, of resources. Mostly they show how global it is, how intertwined it is. And again, they show how you see the kind of repetition of marginalized labor, the repetition of waste which is maybe not necessarily acknowledged. The exhibition ended with The Weight of the Cloud, which was a chapter about all those cat videos we share on the cloud and on our phones, which are an average phone contains 75 elements from the periodic table, so almost two-thirds periodic table. This one work, which I can mention, one of the last works in the show. It's a work by Mary Mattingly, a US American artist who deals with the material flows, literally with supply chains, mapping the supply chains of cobalt. Again, a material which is essential to phones, but is also essential to renewables, it's essential to batteries. We don't think about it necessarily so much, but it's an incredibly embedded, intertwined with our lives. Okay, so now image ecology, that was the history, and the next exhibition I curated, which is now in CR Berlin, image ecology, is in a way for me was a bit about what I learned from that. So if we think of the history, the title of the lecture I said was on fossilized sunshine, beyond fossilized sunshine, on photography's metabolic turn. So the history that we showed in the MKG, in the mining photography show, was the history of photography as fossilized sunshine. And what we tried to do in image ecology, or what I was interested in doing, is kind of exploring, basically trying to just give attention to the things I noticed while working with all these artists in mining photography. And I noticed that a lot of these artists are thinking about image making differently. For me, I mean, in a way, they are thinking about image making beyond fossilized sunshine. They are thinking about image making not only as, let's say, a window to the world, or as Roland Barthes would say, a message without a code, something which is about the transparency and the immediacy of the medium. Rather, they are thinking about it as something which is always in material exchange with the environment. So the question we asked in this show was, what would it mean to think of an image ecologically? And now, ecology is often, I think, we use this word kind of maybe frivolously sometimes. Don't pay so much attention, but the science of ecology, there's one beautiful description of it by Barry Commoner, intellectual chemist who wrote in the 1970s and was very important in popularizing the environmental movement. He described ecology as the science, basically he described it as a science with three main tenets. Everything is connected, just also connected to the talk later. Everything must come from, so nothing is created out of nothing, no? Also a very old tenet of scientific thinking, and everything must go somewhere. So ecology as a science is a science of relations, of change, of exchange, of processes. In a way, it's very much in antithesis to the project, I would say, of fossilized sunshine. It's fossilized sunshine, what photography's great strength, and I think it's also still a strength and still very special, is that it manages to fix, as we said when we talked about Niepce, when I mentioned Niepce, photography, his breakthrough was that he fixed an image. And ecology is all about processes, and the basic tenet is that you cannot fix basically anything. You could call it as the only thing that is immortal is death in ecology. Change is the only persistent value. Now, for this show, what we thought of is, rather than describe or structure the show as according to key materials, we tried, as exhibition makers, to think of it, to take the metaphor of a metabolic cycle. In ecology, often in a relationship of a organism to its environment is described as a metabolism. So a flower metabolizes sunlight thanks to chlorophyll and is able to turn it into nutrients. It emits something which then we can subsist on. This sounds kind of banal, but it's actually kind of radical. And again, if you think of take the proposition that everything is connected seriously, it's very radical in a sense that it means you cannot compartmentalize different aspects. Economy is related to environment. Labor is related to waste. No? So metabolic cycle, the structure of the exhibition goes around energy, material, labor, waste, and back to energy. This is also indebted in a way to Marx, actually, who the same year that, in 1844, the same year that, what's his name, the Pencil of Nature. Pencil of Nature? Talbot, thank you. Yeah, I had too many names. Talbot, the same year that Talbot published The Pencil of Nature, Marx wrote about how nature is man's body, and talked also about a form of social metabolism, basically kind of man's relationship to the environment as one of metabolism and one which is also broken in a sense, or there is a rift that is caused by the extraction, which is integral to fossil capitalism. So the first work you see in this show, going back to Niepce, is a giant heliograph actually, by Julian Charrière, titled Buried Sunshine Burn. It's a very bad image because we can't see the details, but you kind of see this shimmering, weird, abstract pattern. This is also a bird's eye view of an oil field. And it's an oil field outside of Los Angeles. Just outside of Los Angeles there's an oil field. And it's, so now you already know the whole history with Niepce. I don't need to explain. But for Charrière, what he really found interesting is to try and work with the oil from the field, from the neighboring La Brea tar pit, which is an archaeological site where a lot of mammoth and prehistoric fauna and flora have been captured in oil, bitumen, and preserved. That's why there's a giant mammoth. So what you see here, the surface is literally oil. That's one of the first images you see in energy. Second image, which I really can't show you because it's behind closed doors, are these images by Léa Habaudin. These are 5, actually. One is hidden. 5 boxes, and these are antotypes. Antotypes was a technique developed in the 40s in parallel to all these historical photographic inventions that used chlorophyll-based pigment, plant pigment, to capture images. But the problem with antotypes, and the reason we are not all talking about antotypes nowadays, is that they are not fixable. So they fade. for this work by Bourdin, the reason she was interested in this was that Liao has been really interested in ancient forests, all-growth forests, boreal forests, very few of which exist in Europe. And to actually document these forests, you need to gain access, you need to apply, scientists apply for permits. She also applied for a permit. And she was really interested in trying to somehow represent this type of relationship to the forest. So what you see behind these doors are images of forest using the anthrotype technique. I can't show them to you. You'll have to come to the exhibition. But when you open the box, you subject it to light, and you have to decide if you want to open it or not. The viewing of the image, just like the access of the site, compromises its subject. So that's what I talk about for me when I think about the metabolic turn. I mean, this is what I meant about, having learned, or for me, I discovered from photographers, artists, this new way of maybe thinking about the photographic image, but it doesn't only apply to the photographic image, I think. It's really about thinking of how the carrier, the surface, the material substrate is not incidental to what is shown. It is actually essential to the meaning that is conveyed. No? So if you think of photography, as I said, and this is all, of course, it's a bit, it doesn't apply to every case in the history of photography, but photography as a project, if you think of it, what it offered, was very much the sense of immediacy and the sense of fixity, the ability to fix a moment in time. No? Of course, photography itself is also, this is always a sense, there's always an integral kind of denial, because photography itself is also subject to time. And that's why we have to use 50 lux lights if we're working in museums, and very, very specific conditions. But as a project, this denial was integral to it. And I think what's interesting here is then to think, okay, if fading is part of the image, what are the conservatorial conditions that we need to provide it as museum people who run museums, for instance? Do we really need to freeze time? Or maybe not, if that's part of the meaning conveyed. I'm not going to talk about all the projects. We have 3 minutes before I want to open this to discussion. But I think you got what I'm gesturing towards in terms of a metabolic approach. Another great example for this, and I'll say the exhibition is actually diverse in approaches. So let's say a project by Tobias Siloni deals much more with storytelling and text and narrative and fiction, which also have to do, I think, with our metabolic relationship to the environment. So it's not all about exploring this from a material perspective. But what I want to emphasize today is really this question of the kind of weaving together of material and meaning, which might be seen in this metabolic turn, if one could describe it this way. Tristan Duke, in the Glacial Optics series, Tristan is an artist, part of a collective called actually the Metabolic Studio in Los Angeles. So he was part of the show in Hamburg, and this time I showed his work as an individual artist. And he's quite an amazing, also scientific maverick, and he decided at a certain point he wanted to try and create lenses out of ice. So what you see here is a very small glimpse of his Glacial Optics series. Ice, one of the most transparent materials available naturally under certain conditions. And he was particularly interested in ancient ice. If you could think about it, actually, it sounds maybe totally far-fetched and artsy, but this is also quite essential to a lot of scientific research. What you see here on the left are photograms of ice cores from the American Scientific Foundation, National Library of Ice Cores. And ice cores are basically one of our main tools to understand by proxy something about prehistoric or human climate, and also to be able to extrapolate and project onto the future about future climate. So ice cores in their preservation of traces of carbon, in their also various forms of ways they have registered the past, have been a very essential resource for climate, essential resource for climate research. They are between, the ones he depicted are between 14 and 16,000 years old. And they're basically, they have rings, a bit like trees. And what's interesting also about his project and about ice cores is that they are, of course, melting. So in his case, the glacial lens can actually only work when it's melting. Because if it's frozen, if you think of a beer cup, it's iced, it's coated, you can see through it. It has to thaw in order to be transparent. In the case of ice cores, of course, the fact that it's melting means that we are also in this kind of race against time with the main, we could call it photographic evidence of our climate past in our planet. I'm trying to think what is really so essential to maybe discuss. I'll end with my with the beginning. So as you saw, it goes through these different sections: material, labor. You can come to Berlin to CO and see the exhibition and then learn more about each section. One of the first things that now you won't be able to see if you come, because it's winter, is a garden we planted on a small balcony roof in CO. And this is a special garden. It's by an artist called Ulrich Rüter. The work is called Concrescence Plot. Concrescence means growing together. It's also the etymology of concrete. And this garden contains strange plants, a mix of what is called, could be described as ruderal plants. So the type of plants that grow between the cracks in everywhere, in your streets here in Hamburg and Berlin, which Richard collected and cultivated and transplanted into this particular man-made crack. Ruderal plants are, of course, very interesting. They can survive with barely. The kind of description of ruderal plants, let's say, is that they are the first to colonize disturbed lands. So they grow where there's very little nutrients, and they create their own. They can create their own biomass and survive under very tough conditions. So they're actually essential, although often they are also invasive. And the other component in this garden are plants that we selected and could be used for antitypes, for the little boxes you saw in the beginning by Léa Habodan's images. So we actually collected nettle, which is also a ruderal plant, and sent it to Léa, who created those images. And Richard planted some of the same plants which are used for Léa's works or could be used to create these fleeting images of the antitype. So that's how the circle in the exhibition in a way closes, and how we reach also between waste and energy, between nutrients and the basis of image making, the beginning and the end. I think we have a couple of minutes. The plan was to open the floor to a couple of questions, right? But basically there's also later a panel with a Q&A in the end of the day. So if you want, you have a chance now for a couple of immediate questions.
Just very briefly. So thank you so much. It's just a minute. I'm going to go away from the speaker. So I think this is a very interesting way of ending the exhibition because, or like the presentation and the exhibition. Because I saw a video with the artist, with Ulrich Rüter, and it was very nice that he was talking about actually the temporality of an exhibition, We always suffer as curators, I think, when we do something that stays up for 10 days or for 3 months and then just disappears. The only thing you have is a book. And actually it's quite nice to think that you can go back to CO Berlin and actually see this garden growing even beyond the exhibition. And I can say something, there is a little tomato growing just in front of CO Berlin. I think I'll visit it every time when I go there now.
I'm not sure if that tomato survived, but I saw another one that survived. I wanted to tell you. Yeah, now I started looking and finding these plants everywhere. This is what happens when you start taking them very seriously.
Yeah. So now you know what you need to search for when you go anywhere, actually, but particularly to CO Berlin. So we thought it's nice to collect all the questions for the end of the afternoon for the Q&A. But if there's a question for clarification that should be done now, then you're very welcome to speak up.
Wir können gerne auch auf Deutsch oder Ihre Fragen auch auf Deutsch stellen.
Are there any urgent questions?
Okay, so maybe we will digest. Thank you so much, Boaz. And
Welcome back. Willkommen zurück. Viele von euch haben ja schon die nächste Referentin bei der Führung vorhin kennengelernt. Und ich würde jetzt gerne weitermachen. I have the pleasure of introducing our third speaker of our symposium. It's Professor Dr. Regine Henge from Berlin. She's a project leader in the Excellence Cluster Matters of Activity at the Humboldt University in Berlin. She studied biology and obtained a doctorate at the University of Constance in Germany. And after her postdoctoral phase at Princeton University, she started her own junior group and completed her habilitation in microbiology and molecular genetics, again at the University of Constance. She has been a full professor of microbiology at Freie Universität Berlin from 1998 to 2013, and now at Humboldt University in Berlin since 2013. As a project leader in the Excellence Cluster Matters of Activity, she cooperates with anthropologists, material scientists, cultural historians, designers, and artists. Why we have invited her and what connection she has to the work of Katrin Linkersdorf will quickly become clear from her presentation. So we are very happy to have you here. Welcome, Regine Henge.
So, thank you very much for the kind introduction and the invitation to speak here. So, what I want to do tonight is actually to lead you back to where we started this afternoon, that is to the exhibition by Katrin Linkersdorf and what is behind that. So you have seen the beautiful pictures, but there's a long story behind it, and I'd like to basically take you to the lab and tell you what we have been doing in order to make this photography finally happen, and it's really a long story. So I'm going to start by just showing you a few pictures. Most of you have already seen. And I think that these are now a few pictures that really, in a nutshell, tell the long evolution of how Katrin's art evolved. So this is a major piece of the Wabi Sabi series. So this is where she started actually to dry flowers, to expose their crispiness and their beauty. And then, more or less by accident, there's also a story behind it, Katrin discovered that she can actually make these dry plants bleed. So they actually lose their pigments, and she perfectionated this procedure in very long work until to the point that she could completely disdain now flowers. And these are usually tulips. Sometimes there are also other flowers, but with tulips it works very well. And she also then started to actually collect the pigments she extracted and concentrated them them, so basically producing some kind of natural ink. And then she had the idea to actually put it back and recolor basically the flowers. So in principle, color is very important to Katrin. Basically, color symbolizes the life of the flower. So what she is doing here symbolically is she gives life back to the flower, in principle. So this was the state of her art, and as she already mentioned this afternoon, that the way we came together. So she had these beautiful photographs she still has on her Instagram account, and I just came across this by accident, and I was really struck by it and enthusiastic and started to like them and probably also put some comment, I don't remember. And she described it how we came together. So Katrin is very spontaneous. She immediately wrote me a message, and since I was struck so much by these pictures, I immediately wrote back, and I immediately had the feeling that we might fit somehow, yeah? That we have common interests. And so we started to meet. I heard more about this wabi-sabi concept, which is very central to her work, which basically is the Japanese theory of beauty, of finding beauty in imperfection, in transience, and decay, basically. And this is what Katrin enacts in basically her procedure that results in dead flowers, but during the process of dying, they reveal all their inner beauty. And so I was really impressed by that, but as a biologist, I think of dead plant material as a nutrient for bacteria. So I'm a microbiologist, so I have bacteria, microbes in mind. And of course, they can use it as a nutrient. And at that time, when we started to discuss, I was also writing a big article on sustainability and circular flows of matter in nature. And so it immediately came to my mind that, in principle, we could further evolve Katrin's art by actually letting something grow on it. And since I'm a microbiologist, of course, I would like to use bacteria to grow on this dead organic matter of the plants. So basically, to just come full circle yeah, and I should also mention that this circle, which is basically still very simple, it's just death and regrowth, basically, is part of a larger circle, which is the fundamental circle or circle of flowing matter, which basically is the carbon-oxygen cycle in nature, which drives the entire biosphere, basically. We are all dependent on this, and we are participants in this. So basically what happens is that, in principle, all organisms that can do photosynthesis, so everything that's green over there. So it's not only plants, it's also cyanobacteria, which actually during evolution invented photosynthesis. They take carbon dioxide and convert it into living organic matter, into their own matter, basically. And which then, by dying, becomes again food for the so-called organotrophic living beings like bacteria, fungi, and animals, and humans, of course, also. So basically, it's a double cycle. And all living matter or all living beings, they also have an energy metabolism. A large part of it is aerobic respiration, and this means a conversion of organic matter, of living organic matter, or dead organic matter if they feed on it, into carbon dioxide again, which is then again basically the raw material for the photosynthetic organisms to grow new organic matter. So this is the carbon cycle, and it's the fundamental cycle in our biosphere we all depend on. Okay, so the idea was, well, to at least visualize part or the inner circle here. So we needed some sort of bacteria that are able to grow on these dead plants. And this is not trivial, because these dead plants, after Katrin's complex destaining procedure, are basically cellulose. And most bacteria cannot grow on cellulose. Fungi can, but bacteria usually not. So that was one criteria, which is not so simple to fulfill. The next criterion was basically to find a bacterium which is visually attractive, and that means a bacterium that makes pigments. And the third criteria was we had to find a bacterium that not only grows and makes beautiful color, but that is also harmless. So it shouldn't be pathogenic, and of course it should not be genetically modified, etc., etc. So it should be something simple that occurs everywhere and cannot do any harm. Because Katrin wanted to work in the lab, and she has no formal microbiological training, but she saw the lab and said, wow, I want to work here. and so I set out to actually train her as a microbiologist, and now I can say she's really good at that. She knows how to work sterile, and she can do everything now alone. And actually for me it was also an opportunity after 30 years of working in the office, because I am a professor, I have a group, and I tell people what to do, but I don't pipette myself on the bench. So after 30 years I went back to the bench in order to teach Katrin how to handle these microbiological instruments, and I discovered that I'm still able to do that. I'm really proud of that. It's something like biking or swimming, you don't forget that, so that's really nice. So we are both in the lab, and we do these experiments. Now, what are the bacteria that we use? And I had the idea, well, there are certain soil bacteria, they're everywhere out there, they're completely harmless. They are called streptomyces. And these bacteria are actually easy to get, and I know people who work with them, and you can easily handle them in the lab. And, but these bacteria are very interesting for a number of reasons. So first of all, they have a very complex and very interesting life cycle. So when they were originally discovered, it was actually the form that is depicted here in the lower part of the image. They grow in a mycelium, and initially people thought they were fungi, because fungi grow in this kind of large mycelium, but it turned out they are bacteria. And initially when they grow as a mycelium on an organic surface, basically they grow into the surface, but when they run out of food, of nutrients, then they start to erect this what is called aerial hyphae, so they go up into the air, and then this part actually forms cross walls, and then it looks like beads on a string eventually, and then it falls apart, and these are now single cells which are called spores, and spores are a form of bacteria which are kind of dormant and super resilient and resistant against all kinds of stresses. So it's a survival form. So the single cellular form of these bacteria are these spores. And if these spores which are basically blown away with the wind, or they hitchhike on some other bacteria, or they hitchhike on some little animals, like the ones that we also found in some of our experiments, then when eventually they find a better place, then they germinate again, and then they form again this mycelium, and then the whole thing starts again. So this is a model system for complex differentiation and sporulation, and this is why it's rather well studied, at least this aspect of their life. But there is another aspect which is very important for this project, and this is the ability to produce color. And I should also say streptomyces are the major producers of antibiotics. So all of you have at some point been medicated with antibiotics, and most of these antibiotics that we still use today come from streptomyces. Some of them have been chemically modified, but they go back to mycelial antibiotics. And some of these antibiotics are colorful. And especially two of them are well studied or well known. And this is a blue one called actinorhodin, and another one which is red, which is undecyl prodigiosin. And because it's so well studied, and people are actually looking for additional antibiotics coming from streptomyces, we see in the genome that there are lots of gene clusters, unknown gene clusters that look like antibiotic-producing clusters. So there are more antibiotics to be found in streptomyces. So this is why it's a model organism also for studying antibiotics. And this means that we have strains that make either one or both of these colorful antibiotics or none at all. So we can play around, and at least in terms of color, we can predetermine more or less what we see in our experiments. And what you see on the right side is actually a culture on an agar plate, which is very new to impure, and you see this dusty, white, snow-like material. These are the spores. So they produce color and spores at the same time, which is also a visually attractive element. So, but then we had the problem how to bring bacteria and the plants together. So we had to figure out a method to do this. Now, bacteria grow on agar plates in the lab, and we, well, wanted to stay on this system because we wanted to work in the lab. On the other hand, these are the plants. This is how Katrin brings them to the lab, and they swim basically in an alcoholic solution. But alcohol is not good for the bacteria, it would kill them. So basically we have to get rid of the alcohol. So what we do, we wash the plants in sterile water. The plants are really resistant. I mean, you can harshly wash them and do even worse things to them. They're going to survive that on the other hand, we then isolate the spores, because that's the single cellular form that we can easily isolate, put it in water, and then we have the bacteria in drops of water, and we can put it on the plants. And the plants we can put on agar plants, agar plates. And what you see here is the very, very first experiment that we just did for testing. But some of these plates actually made it into the exhibition. That was not planned at the beginning. So these are just petals of tulips, and we put them onto the agar. In other cases where we have bigger plants, and which are also a little three-dimensional, we put them into larger agar plates, and then we pour liquid agar around them, and then to solidify, and then it's fixed, and then we put the bacteria on top of the plants. So basically, the bacteria now have two choices for nutrients. They can grow, or they should grow on, at least they have a cellulase, an enzyme to degrade cellulose, so they should grow on the plants, but they also are known to be able to grow on agar, so they actually can degrade the agar, which is also something very special that other bacteria cannot do. So there's nothing else to feed on in this plate, it's just water plates, basically. So the question was, of course, can they grow? Would they produce the colors? What would they look like when they grow there? And would they, in one way or the other, interact with the plants, so grow differently on different plant parts? We had no idea. And actually, we were kind of discouraged by a colleague who said, well, this is going to be very difficult, and she's a streptomyces specialist. But we said, let's try. And the first experiment worked spectacularly. So these are just a few cell phone photographs from the very first experiments for the lab book. We have a real good lab book also, where we document every detail. And what you see is that this very dark color. So this bacteria grew on the plants, and they produced much more color than on standard agar plates, which is great. In addition, they sporulate. This is when you see when you shine light from above, then you see that white stuff, dusty stuff that are the spores. We could also see that the bacteria were actually able to grow into the petals and actually grow along these vessels, which are these wavy lines that you can also see on Katrin's fairy images. So the bacteria can fill that and produce their color there, so they color the lines basically. So they really, it's like painting the lines in these petals. And they grow very differently on different parts of the plant. So what you see here are the inner organs in the lower row, inner organs of a tulip. And you can see that on one part they grow as spores, so that's the white dusty stuff. And in other parts they grow more on the agar plate, and they produce much more color. So they sense the differences of the surfaces where they grow. So that was all very good, and that was very promising. Okay, the next experiment that Katrin did was using a chickpea, I think. And so she had everything arranged very neatly in the plate and put the bacteria on it, and nothing happened. Nothing happened for a few days, nothing happened for weeks, and even for a month or so. So obviously, and even repeating it, it didn't work. And so we were really lucky that we started with the tulips, because there it worked. Had we started with these chickpeas, then nothing would have worked and we would have given up. So, but then Katrin had the idea, well, peas and probably also chickpeas, they actually make a toxin. So this is why you have to boil peas before you can eat them. And the toxin is called phasin. It's a protein toxin. And well, so we said, well, maybe it's some sort of toxin. It doesn't have to be this one. But let's try what happens if you boil the plant. And then Katrin went ahead and boiled them very harshly. And actually we made a series of different time points. And then it turned out that then the bacteria could actually grow on the plants. So boiling actually destroyed the toxin, which means it's most likely really a protein. And what you also see here is actually details from two of the images you have seen in the exhibition. And this is just to illustrate that the bacteria really grow on the plant material. So on the left side, you see these peas. They are all pretty much intact. And on the right side, which is, I don't know, a month later or so, you can see that the peas are actually being digested and the bacteria really grow on them. Okay. So sometimes the bacteria also really surprise us. So this was one of the examples. So on the left side, which is earlier, the right one is the later one, maybe also a few weeks later. And so on the left side, you see blue and red color. This is what we expected, because that's the strain that has both antibiotics, the colorful ones. But after a certain while, then a new color came up, this brown, rusty orange. And we were really flabbergasted by that, because it wasn't described as another colorful antibiotic. But then I found out that there are certain molecules that the bacteria actually secrete in order to catch iron. Because iron, soluble iron, is very scarce in the environment because of the oxygen, which would immediately oxidize it, and then it precipitates. So soluble iron is a problem for all living beings because we need it. We have it as cofactors in our enzymes. And so the bacteria have very intricate methods to actually send out iron-catching molecules, and then they bind iron, and then taking the molecules back. And this is obviously one of these iron-catching molecules, and once it has bound iron, then it develops this rusty-like color. And then eventually, waiting a little longer, additional actors showed up on the plants, which we had not yet planned, or which we had not put on it, simply because when Catherine takes the photographs, I mean, we have to open the plates in order to allow for really clear-cut photographing. And then the plates are open for a certain while, which means, well, there are certain spores of fungi around and molds, and they can fall on the plates. And occasionally that happens, and then the mold started to grow on the plants. So what you see here is on the left side or on top panel is a white mold, and this is really a killer. So it can eat the plants, it can eat the bacteria, it even eats the color. So after a certain while, there is not much left there. But even more interesting are the black molds, and because sometimes we had examples where bacteria did not grow on the plants because obviously there was some toxin, and here it was actually a tulip which had some toxin, so it didn't let grow the bacteria. And then a mold, the black mold, developed, and after a while, suddenly the colors of the bacteria showed up. So that means the mold had helped the bacteria, so the spores of the bacteria were not dead, but they were just laying around, they were inhibited for growth. But then the mold actually, probably molds are known to secrete a lot of enzymes, secreted a protease that degraded this protein toxin, and that helped the bacteria to then grow. And now this is an interaction which is probably meaningful in soil, where these fungi and bacteria occur normally. And so it's very well possible that after the bacteria grow, and remember they're antibiotic producers, so they produce lots of also antifungal compounds, so they could help the fungus to fight against another fungus or another parasite or whatever, an enemy of the fungus. So they're probably helping each other. And this points to very, very complicated relationships, which are actually so complicated that they haven't been studied so far by science. And, well, here I would just like to show you what this looks like in the lab. So this is a real microbiological working bench. And so Katrin has a workbench in my lab. And this is what the incubator room looks like, and where these plates and what is on them is actually developing. So in the middle you see actually that pea plant, which is now the photographs in 2 versions are hanging in the exhibition. Okay, so finally, I don't have to say much about the photographs of Katrin, so the end products of this long procedure, because you have seen them. and just show these two examples. And this is also a very nice and I think a very special one, because it's probably biologically the most complex one that we have had so far. So as I said before, in the exhibition, it contains the plant, it contains the bacteria, it contains the mold that has helped the bacteria to grow there, and in addition we see these little traces, and these are springtails, little insect that we actually by accident introduced in here without knowing it, because these springtails are actually attracted by the streptomyces, by the bacteria, chemically, and the springtails actually then pick up the spores and distribute them. And this is the trails that you see that. So it's a very complex relationship that you would find in real soil. Okay, so what I have told you so far is actually, you can see that it's not only about the final products, the photographs, but behind the photographs is actually a very long and complex procedure. And this is a procedure of experimentation, of conceptualization, of discoveries, sometimes surprises, and well, moments of wonder when we come to the lab or to the incubator room and discover something that we would not have expected. And this process actually can also be seen from very different perspectives, and this is what I'm going to talk a little bit for the rest of my talk. So basically, the agar plates, and what you see is one of these agar plates here. They're actually big like this, very special agar plates. We look at them as stages where these bacteria actually can perform. And so we see the bacteria as living active matter or non-human actors. And in order for them to perform, which means we basically provide them with a complex system, but we don't set very much constraints. So we just let them do what they want. We just tell them, show us what you're capable of, basically. And so it's always surprising for us what comes out. And what comes out is actually really complex, and it's too complex to be studied by, well, normal science. And it's really amazing. There are just a few people in the world who study streptomyces with its normal ecological interactions in soil, because it's so complex and not much is known about that. And this also means that what we do here on this, well, in an artistic project, also has a scientific value, because what we do here is we allow things to develop that in normal scientific experiments you would not see, because we don't have these constraints, basically. And so that means we go into the unknown. So we don't have a certain question in mind that you usually have when you do science. So you have a question, you have a hypothesis, and then you test the hypothesis, basically. But here we don't have that. We just let the bacteria tell us whatever they like. And so this is an opportunity to discover something really new, and actually, first of all, discover new scientific phenomena and therefore scientific questions. And basically, this can become the starting point of new scientific projects, where we then narrow down and become more stringent in the analysis, which is not what we are going to do, but we are actually going to publish this also in scientific journals and tell our streptomyces colleagues, here are interesting phenomenon, just jump on it and do the research. Yeah? So that's the scientific and the interplay with the art aspect. And of course there is an ecological perspective of this work, because the Microverse project, it actually visualizes and kind of reenacts the processes that happen normally in the dark soil. And I should emphasize that this happens in healthy living soil, because soil is a living entity. It should have a relatively high organic content, and it has a very complex ecology. So soil is living, basically. However, today, about half of the organic carbon in soil worldwide, on average, has been lost already because of agrochemistry-based industrial agriculture. So there's this Monsanto-style agriculture, basically. And the consequences are devastating. So basically, if you look at this scheme again, so what is this kind of agriculture about? Basically, nowadays, we grow plants, okay? We generate living organic matter, but we do not reintroduce the waste, organic waste, basically, into the soil. But on the contrary, we very often burn it. And some countries are totally polluted. If you look at India, for instance, this time of the year, the northern part of India is totally polluted because of burning all this agricultural waste. So the agricultural waste does not go back into the soil. It produces carbon dioxide. And because the soil is actually impoverished, also there are less bacteria. And on top of that, all kinds of chemicals are used, all these pesticides. So we use fungicide, we kill the fungi. We use insecticides to kill all the little animals. We use herbicides in order to kill all other plants. So basically what this kind of agriculture ends up with is kind of a mineralic soil. And the plants just grow on it, and they can grow only because we fertilize them with chemical fertilizer, which they cannot really take up well because they lack all the fungi and the bacteria that normally would help them to actually take up the fertilizer. So this is a disaster. Actually, initially, when this kind of practice started, the soil was still very much intact, and if you add fertilizer, chemical fertilizer to intact soil, you actually get an increase in production. Yeah? But this is for a short time, because then all the equilibrium basically gets lost in the soil, and you end up with this kind of non-fertile soil. And it's not only non-fertile, it also doesn't retain water, and it doesn't retain carbon anymore. Because it turns out that the soil is actually one of the major carbon sinks, and restoring soil, which is not too difficult, but it can be done, restoring soil would actually make a significant contribution into binding carbon, actually excess carbon from the atmosphere. So solving or making a contribution to solving the climate crisis, basically. So basically, there is no other solution than going back to this circular flow of organic matter in soil and the growth and decay that we like to illustrate also with these photographs. And the final perspective I'd like to allude shortly to is the perspective of art history. Now, the circular flow of organic matter and growth and decay is certainly not new in art history. But if you go back some 350 years, we find Baroque and still lifes. And I think all of you know these beautiful flower bouquets in Baroque and still lifes, which are very much reminiscent of, or very similar to what we see in Katrin's exhibition. But, and these flowers, of course, are also in the process of decay. They remind us that all living beings are mortal, including ourselves. So this is the memento mori aspect of these still lifes. In addition, there is a special sort of still life, which is called sottobosco still lifes, which was very popular especially in Holland, but also in Italy. And this sottobosco still life basically show growth and decay in forest soil. So they're soil still lifes. And these still lifes, in a way, they are very ambivalent in many aspects. So on the one hand, they're kind of frightening. On the other hand, they're also very, very beautiful. You can see animals which are eating up other animals and the plants, like these toads. Sometimes you have lizards or snakes, so it's kind of frightening. But on the other hand, you also have animals and the beautiful blue flowers, which stand up for a bright world, so like the butterflies, and the blue is, of course, the color of the sky. And so all this is very symbolic, and especially the meaning of the color. And at that time, soil was really also a very interesting subject for science, which at that time was not called science, it was natural history. But it was a time where natural history was really revolutionized. It was also the time, so 1660 is when this still life was painted. It's also the same year where the Royal Society in London was founded, and a few years later they started to publish scientific correspondence. So this is when scientific publishing was established. And again, a few years later, Newton published his new theory of light in this journal, in the Philosophical Transactions of the Royal Society. And so the old idea of color was the idea of pigments, and now there was also this new idea of color being present in white light coming from the sun, basically. And that actually generated a whole story of color theories over the century, including contributions by Goethe, for instance. Now, I really like this blue color standing for the color of the sky, because when we look at Katrin's image here, or see a look at our bacteria, I think I forgot to mention these streptomyces are called silicolor. So the species is silicolor, which means color of the sky, because they make this blue pigment. So there's this, similarity. And another interesting aspect in these Sotobosco still lifes was that the plants that are shown there are very often medicinal plants. And so at that time, plants and organic materials were, or sometimes also mineralic, but mainly plants, were the major medicinal treatments, basically. Now, medicinal plants, of course, also have this ambivalency. So it depends on the dose, whether they are remedy or whether they are poison, basically. Yeah? And in our example, we also have this medicinal aspect because these colors are antibiotics that can kill on the one hand, but on the other hand, they can heal. Yeah? And so there is a lot of similarity in Katrin's picture and these Sotobosco still lifes. And when I prepared this presentation, I put these two together, and I realized that here is really it's really striking, the similarity. It's not only similarity in concept, but it happens to be also formal and in composition, basically. Yeah? And so if you look at it for a while, you even can see the same structures in the two pictures. And so I leave that for a while. You can look at it, and you're going to discover all kinds of similarities. And so, of course, this was not intended, but I don't know, maybe it's just chance, but maybe it's also because there's an inner relationship between these two kinds of pictures. And with that, I'd like to close. Thank you for attention, and I think we can take questions. Thank you.
Okay, are there any urgent questions concerning this piece, or should we start with a complete discussion? Yes, we should. Oh, shouldn't we? Yes? Okay.
Oh.
Okay.
Then we can.
I don't know if I missed it, but what's the function of the bright blue color and the biological function of the bright colors?
Yes, please.
You mean on the right picture?
Yeah, of the antibiotics also in the other pictures.
So the function of the antibiotic is to be an antibiotic. So basically for streptomyces.
But of the color.
Huh?
But of the color.
Why it's colorful? I don't know whether there is a function of the color. I mean, it happens to be the chemical structure that is reflecting color. And but in principle, there's a very interesting connection between medicinal plants and coloring plants. They're very often the same. So there is this connection between healing and colorness. And I mean, it's not necessarily here the case, even though streptomyces has been used to actually stain textiles also. You can use it as a colorant in principle. And you can do that with many medicinal plants. Indigo, for instance, is an example. So it's a medicinal plant and it's a de-stain colorant plant. And I think the reason for that is that very often molecules that are colorful are oxidized molecules. That means they tend to lose electrons, and then they go into the colorful form. And the electrons have to go somewhere, so they go to some other molecules that need an electron. And molecules in need of electron are themselves oxidized or can be oxidatively damaged. So for instance, we all hear very often about oxidative damage as a cause of aging, for instance. Yeah? What that means is basically electrons are withdrawn from our proteins, from our lipids, from the membranes, and so on. So these molecules, in order to be repaired, they need electrons. So colorants, certain colorants, when they go into the oxidized, colorful form, they give off the electrons and they can give it to basically oxidatively damaged molecules. Heal the molecules, basically. And I think this is the connection.
Here's one more question.
Oh, yeah. Yeah, thank you very much for it. It's a really good understanding story. And I want to ask you, what does all this work mean for you, for your future work? Could you tell me this?
For my future work, well, I'm actually closing down the lab in 2 years. And during these last 2 years, this is a major project going on in my lab, so we're going to continue that and develop it further. For me, I already mentioned a few, the value that it also has for science, so that we actually discover phenomena that nobody else has discovered so far, because these are very complex settings, and that are a source of new scientific questions, which I will not solve, but I will discuss that with colleagues who have more time to further work on that and who might be interesting to solve it. And in addition, when I was introduced, it was also mentioned that I work together with lots of people, colleagues from other disciplines like anthropologists, like material scientists, even art historians and designers. So I also have designers in the lab. So I used to be a molecular biologist, but I'm getting out of this, and I'm getting more into this interdisciplinary kind of work. And basically the headline that I could put on top of my research currently is, I'm interested in complex interrelationships in nature, so for instance between plants and microbes, and in order to understand these interactions, and then we can actually use it for solving our own problems. So we have to get away from solving problems by just using chemistry and inorganic whatever materials. We have to go back to organic materials, organic processes, in order to basically reintegrate our activities into the biosphere, basically. And it's very important that we understand these complex processes so that we learn how we can use them, basically. And one way to learn more about these potentially interesting procedures is actually also to look into traditional practices. So, for instance, I have a project going on with an anthropologist. She's working on dyeing textiles in West Africa using very complex mixtures of plants and earth, mud, which of course contains bacteria, and the bacteria and the plants interact, and we find that this is very important for the dyeing success, basically. And this is a possibility of changing dyeing processes away from toxic chemicals to more organic molecules. And there are lots of dyes and colorants in nature that we can actually use for that, and we just discard it. Yeah? But we have to understand it better to control the processes and actually use them. Thank you.
Yeah, thank you. So we should open the discussion with all.
Okay. So, yeah. Was not intended for me to sit in the center, but it just happened organically. So, yeah, thank you so much to all of you, also to Katrin, for starting the day in her exhibition and for actually, bringing us to have this symposium here today. And also to Boaz for making a short introduction to a very, very long research, to Mathieu for sharing a raw project. Like really, I need to really point at that it's, I feel it's very hard for curators and artists and whoever, like scientists, whoever is in the middle of a process to actually open it up to an audience. I think it's very, it has a lot of value, so thank you for that. And to you, Regine, because I think it really showed, like to me, it really closes up a cycle about interdisciplinarity in the arts and in science, and how much we need to listen to others and actually leave that bubble of photography quite urgently. So I really want to open the conversation for the whole audience. This should be a horizontal conversation. We've spoken enough. But I have maybe just one question to open it up. And it's the question of, let's say, outreach and accessibility. Because we've been speaking about exhibitions and about artworks, and I've seen, like, I brought these 2 photo books or 2 catalogs, let's say, that are actually the result of the 2 exhibitions that Boaz presented. The exhibitions and the artworks, they have hopefully they have a long temporality, and their life is not as short as in the exhibition. So from your experience, and I really want each of you to answer briefly, also Katrin, how do you use the tools you have to really make these ideas, this information accessible, and what has been your experience so far given the formats that you've used, so exhibition, artwork, scientific study? Maybe I start with them.
Okay. We actually are doing a book right now for this exhibition, and it's a little bit delayed due to also the status of my photographs, because they are not ready yet, as I explained before. And I think this book, everybody's asking for it. It explains a lot. And there are also essays in it from Professor Henge and also from Karin Leonhardt, who's an art historian. And I hope that brings it in a certain kind of language to people that is understandable. That's not, as we just experienced, Regine is talking quite not in a scientific way so that nobody can understand it, but more approachable. And I think this is one way, and the other way is the exhibition that I have here now, so that we have tours, guided tours, and people ask questions that are regarding to the Microverse series and also to the other ones. And I hope that the awareness, or that as we play with bacteria here, becomes a bit more, not that we think, oh, bacteria are dangerous and stay away from it, because I think that they are a great opportunity to solve our problems in the future, because now we are choking on waste and rubbish, and this is not a cycle of nature anymore. So I hope this will bring it a bit more forward.
Mm-hmm. Thank you.
Yes, I can underline this, because people coming here to look for the unbelievable beauty, and after a while, they're starting asking questions. And this is the idea behind just talk about ecological things and the bigger things behind. So it's very tricky to do this in this form. Thank you.
You want to respond?
Okay.
You might have a very different perspective on it, so I would be interested to hear about, the kind of outreach that you've experienced in the last years, perhaps.
I have been interested in what we now call outreach for quite a while, and not only in exhibitions, but also using exhibitions. Another project I had actually from 2000, when was it? It's almost 10 years ago, but it lasted some 8 years. I had a science and theater project going on with a professional theater, where we actually discovered that in the English-speaking world there are lots of science plays, and which about topics like responsibility of science and science and social interactions and how science is being made, what happens in labs, and what's the human factor in science, these kind of things. So really interesting topics, and some of these plays are actually very good, and so then we started to put them on stage, so with professional actors, a professional director who is a good friend of mine, and that was also a very, very successful project. So that really brought me into contact with outreach, long-term outreach projects. And then in the excellence cluster of matters of activity, making exhibition is actually for us now also even a research format. So because an exhibition is not something that you put there and then it's final and people look at the images or whatever you exhibit, but for us this is something which is in flow, and making an exhibition involves a lot of research, and you have to become clear about what is behind what you want to exhibit. And what we exhibit in matters of activity, mainly in the Tieranatomisches Theater in Berlin, is not so much end products, but we visualize and we expose the process. And the exhibition itself is a process which can still change over the course of the exhibition. And it's for learning. Everybody can learn that, not only people who go there, but also we learn a lot by making this kind of exhibitions.
Thank you. So I think it really resonates with this side of the stage, because you've talked about it before, Boaz, that the exhibition in Berlin, for example, it was more like showing experiments of artists trying to make things visible, and also you have to deal with also this kind of experiments also with the ink, and like all this process-oriented research is becoming very, I think, very important in this kind of ecological exhibitions. So, Mathieu, from your perspective, exactly the same topic, outreach. You've said you've worked in exhibitions, you've worked in photo books, you're working in a new dummy, and also I think you both interact in a very horizontal way with other curators and artists and other experts. So how do you experience this process of interdisciplinarity and outreach?
Yeah, I think the dissemination of the work is quite important, and mostly with the subjects I'm working with, and so I mean, the multidisciplinarity, of course, it helps me to build these different layers in any story. But for me, what is the most important, I think this is what you're asking, what happened with this work when it's done, and how this work can, of course, go beyond a book or an exhibition. So for me and with the curator I work with, Sergio, what is very important is how can we make accessible the work, and how this work, it doesn't only stay in the places where it should be. There are festival museums, but at the same time, how can we make it accessible ourselves, so the work can be in a small public library in the middle of nowhere, in a school. So, yes, I think for us, and as I always say, we have what we call this kind of champagne and caviar exhibitions. There are these huge exhibitions for museums, but at the same time, if you have no money and you want to show the work, and you can, we send you the PDF, you print it as cheap as you can, and so it's that compromise. It's not only about showing in small places, it's not only about showing in big places, but it's about having the tools so that can be possible.
Thank you. Maybe Boaz, you want to
yeah,
finish the round?
Exactly. I'll try and add something to all these good answers and interesting reflections on this question. I mean, for me, I think these two projects have been a bit different. but they share the same, my same sentiment and input. I'm generally kind of inclined not to try and speak to an audience which is not restricted to, let's say, the kind of art world, and that starts from the language one uses, and that ends with, I guess, also with the type of partners you seek out. So for me, and this is, I think, also thematically connected. So, like we said, I think this was a refrain in the conversation, that the type of subject that we're dealing with also requires, it can't be compartmentalized, it can't be restricted to kind of, let's say, ecology, like the environment, strictly speaking, as something which is just one discrete subject. It really requires partnership and dialogue with people from many fields. In mining photography, this meant that we really approached a chemist and what did we have? A chemist, an archaeometallurgist, a biologist who was actually a wetland researcher, one art historian too, and an activist coming from a kind of organization that advocates for supply chain reform. So kind of trying to seek out these partners who each have their own network then, and have their own audience and have their own conversations, and bring those conversations then back also in the form of audience, viewership, etc. So that's always interesting, and it's, I think, very satisfying. And the afterlife of this exhibition has been a bit of a surprise in a way that, we didn't plan it this way, that the book has been really as material for a lot of seminars, it's been just kind of, people take it and use it as a semester to teach this specific aspect of the history of photography, and that's been very satisfying to see, and not something we expected or planned for. And with image ecology, it's similar. We see a lot of, an exceptional amount of school classes coming to CO. In CO school, until up to 18, people get free entrance. So that's another kind of maybe incentive. But they've reported, I can't really, I don't know, I haven't been there for a long time, but there's been an exceptional amount of school classes. So that's been really interesting to see. But it's kind of the result. I don't know if, I didn't come in planning it's kind of with outreach in mind necessarily. It came more out of the theme and out of the questions, organically, in a sense. What else can I maybe to add about this? It was also really, I think what was important for us was a bit, this goes back to the question of process and research, to change the type of conversations maybe that are often being had about these questions, which often these questions are kind of, maybe obscuring the process and looking at results, of looking for solutions rather than relations, and looking for solutions rather than causes. And those were the type of things that were important, and I think this then, builds on that.
Mm-hmm. Thank you. So just as a little reflection and also a learning for myself in arts and in curating and creating artworks, we think about the material, about the matter. That's become quite clear. So when you ask yourself how can you do an exhibition that is kind of ecological, like environmental friendly, I was actually quite positively surprised. What can you do with a book? And it's when I encountered the catalog of image ecology, I realized that the cover looked a little bit weird, and then I saw there are very different covers. So it basically uses, like, the dust cover is basically paper that normally would be thrown away. And in each of the chapters you find really like lots of information about what the paper is, where it comes from, what the ink comes from. So how can you use something that is expected in the arts, like a catalog, to really bring the conversation farther? And I read the text you will have in the catalog is also amazing, and how can you expand that conversation? So yeah, good examples there. Here it's my 2 books, so don't take them home, but you're very welcome to look into them. So maybe we open it up so the time is not immediate.
You can even ask. You can even ask in German if that's easier. Then we will translate it. You know, just in case. If not, I have a question that I would love to ask in German, but I try it in English, especially Mathieu, but to you other guests too. How can you avoid to be part of the system by yourself? For example, Mathieu, you mentioned that many photography prizes are sponsored by business that is problematic, and even your artworks, I saw them, they are amazing, but how can you avoid that the people who bought the car will buy the artwork relating to the car? And especially for institutions like the CEO Berlin, there's one exhibition that deals with ecological aspects, but there are 10 others that come from Japan or moved to, I don't know. So it's always difficult that you are not part of this greenwashing by yourself, so to say.
Yeah, this is a very interesting question. And until now, for example, my work of Monsanto, I think before apologizing myself in some sort, because this is pretty much what I will do, I think the most important thing is to understand that we need to be very careful with the individualistic guilt. And we need to understand that that works very good in the sense we, each one as individuals, are responsible. That doesn't work like that, and this is a very good way to put us apart. And we're not responsible for that. You as a consumer are responsible for that. So we need to be very careful with that. On my side, what I can say is I'm a child of capitalism, as we all are. I enjoy capitalism. I live in the first world, like what we are. I'm very lucky. I'm glad to do it. So yes, I buy into that. Nonetheless, the little things I can do to be coherent with my narrative is put certain tools that will keep me on this ethical process. For example, my work of Monsanto, I decided that is a work that I won't sell. I won't sell to private collectors, but not because I don't like private collectors. I love private collectors, but because the work is not made for that. What happened with one photo of Monsanto? It doesn't say anything. So, you know, all these strategies. At the same time, the gallery that asked me to be a part of the gallery, I told them, sure, I will be a part of your gallery, but you're not going to sell the work of Monsanto. And the first exhibition on the gallery, you need to stop to be a commercial space to become an educational space. And again, the Deutsche Börse, when I was nominated for the Deutsche Börse, I made an exhibition that denunciated the Deutsche Börse. Of course, also you risk to follow on the trap of they absorb that. You become institutional criticism. Institutional criticism can become a part of the what you are critiquing. So it's very complicated, and there is not an easy solution. This is why I think for me the best solution is to have a job as a teacher that give me enough money that allow me to, no, I'm not going to go in this way. No, I'm not going to do this. But again, I'm a, you know, I don't have kids. I don't have, so for me maybe it's easy to navigate this as better as I can. And I know that it can be more or less difficult depending on who you are, what you do, and all these things.
Can add from my perspective, because you mentioned institutional. So as an institutional, I can't yet speak so much as an head of an institution because it's been only 3 weeks. But of course, these questions will come to haunt me in the coming years, I'm sure. No, but I think on a very basic level, I didn't go into it so much in the talk because of the brevity and trying to keep it short. But when I showed this goes back to what Mathieu just said, when I showed the slide about the Anthropocene show, which was very well known by Buzinski and others, curated by Ustal. There are kind of two ways to describe the origins of the climate crisis. There's several ways, but everyone agrees that it started more or less, let's say, the acceleration of carbon emissions and the ruination of planetary systems started around the same time. But what you ascribe it to is hotly debated, also in circles of environmental historians. One way to describe it is to call it the Anthropocene, and that implies it's the age of man, and that means that humans are somehow equally responsible for what we are seeing now. Although actually for 99.3% of the existence of the human species on the Earth, we didn't destroy the planet so effectively. So that is, and actually geographically, as Mathieu also described geographically and in terms of the specific origins of the climate crisis, it's actually a very geographically limited and small, it's a very historically specific phenomenon, and we often forget this. It's not all of us in this together, we are all suffering the consequences, but the story of carbon footprints as something individualized, which is really the very strong studies showing how oil companies have used this narrative and propagated this narrative for the benefit, is like the biggest distraction from what actually has to be done. One statistic one could say is, there are 100 companies responsible for 70% of carbon emissions. Another statistic that one can talk about is that the wealthiest 10% of the world's population are responsible for 50% of carbon emissions, while the least affluent 50% are only responsible for 10% of carbon emissions. So you have a very, very dramatic distribution of responsibility which should be addressed. And I think the last thing we should do is ask museums, what are you doing to solve this? Because actually we should be asking politicians, what are you doing to solve public transport? And what are you doing to instead of encouraging the use of private cars, for instance? And these type of conversations. So I think it's very dangerous to fall into that trap, which we do again and again, as if we can buy ourselves out using, organic, kind of carbon compensated products, which unfortunately we can't.
Yeah, I think what you said, Katrin, you wanted to say something, no? Sure. Okay. It really, with culture, it happened, I felt like during COVID, culture was actually the first thing that, was closed up. And so we became unnecessary in a way for a little bit, which happened actually to be a bad idea, because gathering together is actually what can bring us forward in such a thing. But I remember in Hamburg there's been a study, museums got together to actually study what is the carbon footprint of the museum landscape in the city, and you can imagine that also a major part of this carbon footprint is actually the tourists, the travelers that come here to see exhibitions. But when I talk about this with the former director of Kunsthaus, I was saying it's so good that they are starting looking at that, and she actually stopped me and said can you please look over the river? And I forgot, we are looking at museums in Hamburg and we're not asking what does it mean to have the harbor, what does it mean that there is no diesel, all the things. So as you said, it's very, we need to be very careful. That's the...
I wanted to add something very quick, and in the sense that we are all a part of this system, and this is in the sense that we all depend on growth, and I depend on having more exhibitions, I depend on having more in this kind of growth, do new work, have different exhibitions in different places, teach everywhere where I can, and all this, my economy depend on this. Of course, more and more I try to say, like I came from Arles, for example, to give, to be a part of this conference. So it's a very complicated problem, and we are all in this problem, and I think it's a political power, of course, through us, to dictate what are the changes necessary. Actually, and to finish, it's very interesting what some theologians are talking about, that maybe, and we're start to seeing it in China, for example, maybe the only solution for this is a ecological dictatorship. What is a quite interesting way, approach to ecological solutions that is completely crazy, of course, but I mean, you understand on the mess we are today, and I think, yes, it's very hard to yes, just take your bike, or just take or say no to a job, or it's quite complicated.
There's a very interesting website, the Degrowth Toolbox, if you want to look at it online. It's actually a very nice guide for how can we use degrowth within arts to actually, and it's also about care, and it's about many concepts and the buen vivir, so it's really not about the, it's very practical to look at it, to learn more about how we can act. So I looked into the audience, because we are
almost finished,
okay.
But not really.
No, so...
but, okay.
No, I just want to look if there are questions. I mean, I know this moment, it's always a little bit awkward. We will have a moment to talk afterwards, but yeah. Carlotta?
Thank you. I have actually two questions for you, Mathieu. I think the other ones can say something to it, but maybe it's too specific. I would be interested in concerning the Monsanto project, if you could give a little insight to your research process. You briefly mentioned that it's a lot of time spending on the internet, but yeah, how much time, or do you also have like scientific works of researchers that wrote papers on Monsanto, or other people that you got to know along the way of realizing the project? Yeah, and I think the second question I'm going to ask afterwards.
So yeah, when I say a lot of hours in the internet, it's because pretty much all the research that would lead you to meet people, to understand who are the people who understand the subject, the scientists, the lawyers, the activists, you need to go through the internet first to see who they are. But of course, I work with the Center for Food Safety in the United States, in Washington, D.C. This organization gives me access to the farmers who are being sued by Monsanto. So how much of the research? Sometimes too much, in the sense that I need to find this balance between getting out of this research mode that I start looking a little bit like a hamster, and put it on practice and come back and go and come back. So, but yes, the research takes a lot of time, and what is beautiful also about the research, and I think it's very important, is allow yourself to get lost. And allow yourself to suddenly you end up with a video of a cat in YouTube. And, maybe that would lead you to other things. But yes, I think the research is a very dangerous thing for a photographer. But if you kind of control it, it's quite a wonderful thing. And yes, how you're going to photograph, why you're going to photograph like that, you're going to use film, digital, black and white, color, all these and more practical answers. Yes, I think the research for me is a quite important tool.
Yeah, thank you. And concerning facing power in documentary photography or photojournalism, have you ever faced backlash or kind of restrictions from Monsanto itself? Because a few weeks ago, a fellow studiant of mine and me had the overambitious idea to do kind of such thing on a company in Germany, but we found ourselves, or we think of it as kind of a David versus Goliath situation where you fight against like a big company that also has a lot of influence on economics, on politicians, on anything. So have you ever faced backlash, restrictions, repression?
No, never. I think they have a bigger fish to fry than me. I think, Bayer, they lost something like 40 billion euros when they bought Monsanto, because not only lawsuits, that this is not really the point, but because the potential future lawsuits. So they know who I am, of course, they know my work. I mean, they know who I am, they know my work. But I think, no, they're not too worried. No, I'm not a big problem for them. Now I don't know what will happen with this new work and using the images of all these very powerful car makers. And yeah, and I need to admit, I don't want to get a lawsuit from Bayer or from Renault or Peugeot or BMW. But at the same time, if you're not pissing off people, I think, for me, you're not doing exactly what you should be do. At least for what I do. There are many ways of doing it. But for me, yes, I think it's important. I mean, pissing off in the sense of, we don't like very much what this person is doing. So, it's this balance between, but at the same time, it's very scary. I mean, they can freeze your work and you cannot do anything with your work. So, but I think I count on first, the bad publicity that will give them, and the second one, that they just don't care about me, even if that hurt my ego.
Okay, thank you very much.
So, any other question? I do have a question for you, Regina, because you are the only one on the stage that is not particularly in the scene of photography. And because we were talking about images and impact, I would like to ask you for your opinion as an exhibition visitor and as a listener, what you think, what images can actually impact us or actually reconnect us with that world that you've emerged in, but we're not. So what is your opinion on this? Which images should we keep using to actually engage people? In your opinion, really personal one.
Yeah, that's actually the question. How do you attract people to exhibitions? Yeah. So don't expect me to have an answer to that. But there are related questions. So, for instance, how do you attract people to do things they originally don't want to do? I mean, so at least they have to get up and come to an exhibition, so they have to overcome their laziness at least, so you have to make them motivated to go there. And it's even worse if they have to change their behavior. How do you motivate them to change their behavior? I think one thing that doesn't really work is using images that just exaggerate the what's going wrong, basically. So what works much better is to frame it in a positive manner and to use some humor and actually to try to make it cool, what you show and what you want to, the message that you want to get across. Yeah? I think that psychologically works much better than just exaggerating. Yeah?
Thank you. So, as a connection to that, and I look at my, you also spoke in one of the lectures that I saw on YouTube that I can recommend, talking with an artist. You also talk about metaphors, and like it was an artist. It was in the. I'll remember that and tell you later. It's online. But you talk about metaphors, and I think, like in science, we're very thankful that there are metaphors, so we can actually understand what's going on in the labor. But also, it takes me to the topic of fiction, because I've seen, like, we've invited people that are showing artistic practice that is really not this photo documentarist strategy, but using all the things such as the material research, but also fiction. So maybe you, I know you have works in image ecology, and I have been there with my students. And there are these tricky things of using, like you're showing actually something that looks documentary, but you're inventing a story to get to a point. So, what about fiction? Can we use fiction to drop people?
I'm always, first of all, I'm always a fan of fiction. Also because I think it shines a light on, interesting light on what we understand as documentary, which is often, anyway, interwoven with various forms of fiction, from just the structure of a language, which is kind of, based on metaphor, to a higher level. We had a work in mining photography. There was a beautiful work by Tobias Ziloni, who was talking here last year, which, in both shows we had works with fiction. In mining photography, we had a work where he studied he went to the site that used to be the factory, a film factory in East Germany, Orvo, in Bitterfeld, which was a site of kind of quite a well-known environmental catastrophe. And what he did there was basically realize the only thing that remained from that factory, from that company, from Orvo, was a company that now produces microfilm for archives, including the German Bundesarchiv, for the Vatican archive, because they believe that this microfilm will survive way longer than digital images. And they have a ISO patent. It's supposed to survive 1,000 years. And that was the origin, the starting point for Tobias's work, where he interviewed some of the former workers in this factory that doesn't exist anymore, in a city in East Germany, not so far from Berlin, which doesn't look like, it was used to have a population of 40,000, working force of 40,000. Now there are like 20 working in this one company. And he used the film that's supposed to survive to try and tell a story to future generations in 1,000 years. What will we say about what used to be here, about this former film factory, about the site, about the workers who used to work there? And I thought that was a really interesting proposition. Both because I don't know so many documentary works which go into this realm of fiction. Reminded always of Le Jeté, like Chris Marquer's film. And because it seems so necessary. Because it's like, yes, that's what we need to think about, this horizon of time which exceeds our human, our historical perception of time. So that's just one example. I think it's very, very, very useful to keep thinking also in fiction, because sometimes, the type of connections, the type of stories we need to tell are just exceed what we can show necessarily, and that's a bit of the story in both exhibitions in mining, especially, that it's a history of photography not through what it showed, what photography usually we think of as something which depicts the world, but actually through what it obscured. And what it's obscured is the labor or the waste, and to show that, maybe we need to show it not directly, but through, like in billiard, indirectly. And that's, I think, fiction is a very good tool for that.
Yeah, I think there is in documentary photography also something called speculative documentary photography, and it's of course about fictionalizing, but also about, for example, how can you talk about things that didn't happen yet, but that you can put yourself as a narrator of future events or events that happen already. How can you replay those events in photography? And so yes, there is a type of documentary work that, and Max Pinkers, for example, he works a lot on that, on speculation, speculative documentary.
So I think we're getting closer to the end. So just as a reflection, I think what we need to work is on a different kind of imagination, let's say, to be able, through all this different strategy, to get to a different idea of what could actually be the future, or what imagine for us to be the future for maybe the ones coming. So I want to thank the audience for being so patient and spending the afternoon with us, and I thank you to all of our guests, from the exhibition to the space for sharing, and the conversation continues outside. Yes. And Ulrich,
our
please.
Program is not finished, so stay with us. We just have to move to another place to go to the House of Photography. Yes, in a few minutes we will meet again. It's another get-together. We will show our Sammlerstücke. Also es beginnt jetzt der gesellige Teil des Abends und ich würde mich sehr freuen, wenn alle dabei bleiben und wir gemeinsam was essen, was trinken, uns dann auch die Sammlerstücke anschauen und wir uns als Freundeskreis auch nochmal uns gegenseitig sozusagen kennenlernen. Und das ist natürlich dieser gesellige Teil ein ganz wichtiger Punkt in unserem Programm. Hier schließen wir jetzt. Ich würde auch alle bitten, ihre Garderoben mitzunehmen, weil hier muss alles raus sein. Und drüben sehen wir uns dann gleich weiter mit dem nächsten Punkt, wo wir die Sammlerstücke vorstellen. Euch allen nochmal vielen Dank und alle, die jetzt nicht mitkommen. Wir sehen uns wieder im nächsten Jahr mit neuen Programmpunkten. Aber ich bin natürlich, oder wir freuen uns natürlich alle sehr, wenn ihr alle gleich drüben im Haus der Fotografie nochmal dabei seid. Also, once again, great thank to all the audience, lecturers, and yeah, thank you.
Thanks so much.